Welcome everyone. So we're going to begin today with a presentation on the neuron as the basic building block of the nervous system by our music research team lab manager and honors student, to graduate from Marymount Manhattan, Keaven Caro. And come on in, welcome, sit down. And then afterward, we'll talk a little bit about our music research team, which certainly invites students from the neuroscience club, the psychology club, the biology club, and basically anybody who might be interested in this kind of research. And we'll probably begin with a formal project in the spring, but in the meantime, we'll welcome new members and do a little discussing of some of this material. So, without any delay, Mr. soon-to-be Dr. Keaven Caro.
Hopefully. Hello, everybody. Thank you all for coming. So, as mentioned, I'm just going to give a pretty brief lecture on the neuron and why it's so important. It's one of my favorite subjects, and it's really what made me fall in love with neuroscience and want to pursue it further and switch from dance to science. And I'd like it to be a conversation as well. If anyone's interested or has any questions while I'm talking, feel free to jump in or raise your hand or anything. I'd love to chat with you. So, I guess the big question is, why do we care about the brain? And why do we care about the neuron? So the brain is so important because it really makes us who we are. Its very complex structure, architecture, and function allow us to sense the environment, perceive it, and create our own internal perception of the environment, and it allows us to manipulate it and interact with others. And the brain could not be able to do any of this without the neuron, which is just a single cell that communicates with other neurons and is supported and communicates with glial cells to create this complex system. So I'm going to go into the details on the neuron so that we can better appreciate the brain and ourselves. So the brain is composed of these specialized cells called neurons or nerve cells. And they can be seen here in a Golgi stain on the right, which shows a cell body and all of these projections. These specifically are pyramidal cells, and here you can just see a generic multipolar neuron. And you can see how it makes up the brain. And like I said, this is my favorite subject in neuroscience. So the structure of the neuron is critical for its function. And one of the most important things in biology is that often the structure dictates its function. So as you can see here, you have the cell body, which houses a lot of the machinery to create what the neuron needs to send out signals and to support itself and to survive and live. And coming out of the cell body are these projections called dendrites. And these structures are like the antenna of the neuron. They have many receptors to receive signals from other neurons and cells to affect how this neuron will signal and how it will affect other downstream systems. And inside the soma as well is the nucleus. And the nucleus is very important because it houses all of the DNA that the organism has. And this DNA provides the information to create the building blocks of the neuron and to allow for all of its functions. And here you can see another projection that is separate from the dendrites called the axon. And this axon is critical for sending information over long distances very quickly. And on the axon is a structure called a myelin sheath. And as we'll get into more detail later, this axon kind of acts like a wire, and it's able to transmit electrical signals down the cell. And this myelin sheath acts as an insulator. So it helps speed up the signal by preventing diffusion and loss of the action potential, which is the electrical signal that gets sent down. And I'll get into more details later of that. And so down here, the axon will end into terminals. And these have so many different names. My favorite name for them are presynaptic terminals, but they can also be called terminal buttons, terminal feet, etc., etc. And the axon terminals play a very critical role in communication between neurons. And these structures are very important for the formation of a synapse, which is a little gap between neurons that allow for chemical signaling and therefore allows for the functioning of the brain through the complex processing of information. And this isn't always mentioned, but another thing that I really enjoy is the part where the cell body, or the soma, becomes the axon, and right here is called the axon hillock. And this part of the neuron is essential for creating the electrical signal that gets sent down the axon, and it plays a role in surmising all of the information that it receives, because as you saw in the previous pictures, these neurons will form a lot of connections with each other. And so it's receiving thousands of signals at the same time, and it's able to integrate all of that information to decide whether it should increase its firing or decrease its firing or stop it altogether. And the axon hillock is critical for that summation and the initiation of the signal. So what kind of signaling is done here? So I mentioned electricity, I mentioned chemicals, and so really the signaling of neurons in the brain are a combination of these, and it's called electrochemical signaling. And so the electric portion is done within the neuron itself. So to get information from all the way at the top of the dendrites down to the axon, the neuron will use electric gradients. And so I will get into more detail in this later, but the information received by the dendrites can create a more positive charge in the neuron. And if this reaches a threshold and it gains a high enough charge, it can send this positive charge down the axon like a wire. And so, as you can see here, you can see all these different signals coming in. And the action potential, which is the electrical signal being sent down the axon to communicate with downstream neurons. So, the action potential is the electrical portion, but the chemical portion is from these chemicals synthesized in the brain called neurotransmitters. And these can be proteins, short proteins, large proteins, and they can also just be chemicals. And these all have different categories, but I guess the most popular one are the like dopamine and serotonin and acetylcholine. And these all share very similar building blocks that allow them to be made. So they can range from moderately large to very small, but they have very big impact on how the neuron will communicate with others. So neuronal signaling, as I mentioned, is done within itself via the action potential, and it communicates with other neurons via the neurotransmitter. And as mentioned, these neurons can hear these signals by receptors on the dendrites. And they can also have receptors on the cell body as well and the axon. So it can receive signals at many different portions of the neuron, and they all have specialized receptors that are specific for certain neurotransmitters. So you could be receiving neurotransmitter information from acetylcholine, let's say, but if you don't have the receptor for acetylcholine, that won't affect how the downstream neuron, the one receiving the signal, will function. And so a very easy analogy for this is a lock and key model, where there's this very specific shape in the receptor and very specific shape in the signal that will bind to the receptor and will cause transduction of the signal, or changing from chemical to electrical. And this action potential, in some neurons, it can just travel down the axon, just like, in a straight line. But an interesting phenomenon is saltatory conduction, which is allowed for by these myelin sheaths mentioned earlier. And this allows the action potential, the electrical signal, to seem to jump along the neuron axon. And this greatly speeds up the signaling. And the reason why it seems to jump in this way, it's not actually jumping, it's just preventing diffusion or spreading out of the electricity from the axon, so out, and dispersal, which would require constant reconstitution of the electrical signal to prevent it from decreasing. Because a very important aspect of the action potential is that it fires in an all or none fashion, which means that once it reaches this minimum threshold, it will send at the same strength no matter what. And so if you did not have this myelin here, the signal would start to get weaker, and the axon would have to regenerate the signal as it travels. But because you have this insulation, the signal only has to regenerate itself at these nodes of Ranvier. And so this allows much quicker signaling. And without this myelin, neurodegenerative diseases of the myelin can lead to very awful consequences. I believe amyotrophic lateral sclerosis is one where the myelin sheath gets attacked by the immune system, and it can eventually result in paralysis just because the signal is too slow for it to properly function. So I mentioned diffusion, and I have a very short video on what it is. And it's basically going from a very high concentration, very compact, to very spread out. So you can see here ink that was placed in water, and you can see that it grows over time. So there isn't more ink coming out of nowhere. It's not increasing in size, it's just spreading out over time. And this is just due to the random motion that's inherent in all particles. So if you place it in the center, it will have a lot of ink particles compact together, and because everything has a temperature and a natural amount of kinetic energy, it has vibrations. And these vibrations will cause the compact particles to collide and bounce off each other. And so, just because of random chance, as they continue to collide, they will begin to spread out so that they experience less collisions. And that's why the ink was able to increase in such a great diameter. But you can see that it became lighter as it spread out more. And that's because the particles became more less concentrated. And so this happens in the axon during the action potential as well. The positively charged particles will want to spread out, but the axon will need to maintain this positive charge to be able to send the signal at its correct strength to then trigger the release of neurotransmitters. So, diffusion is going from an area of high concentration to low concentration. And this happens inherently among all of matter. And as I mentioned, there are specific ion channels or specific proteins that allow the positive charge to enter into the cell. Because this outline of the neuron is a barrier from the outside called the cell membrane. And this membrane is selectively permeable, which means that only certain things can get in and only certain things can get out. And without these ion channels, these special proteins, the positive charges cannot get inside of the cell to regenerate the signal. So those ion channels, as I mentioned, are concentrated in the nodes, which allow the signal to be regenerated there. So the reason we know about the action potential is because we're able to use electrodes to measure the difference in potential across the membrane. So what scientists and researchers have done is they take very small electrodes and they place it either inside the cell or right outside a node of Ranvier. And what they can do here is they can have it in reference to the general extracellular environment, and they can measure a difference between these. So naturally, the neuron will have a slightly negative charge compared to the outside. But as it receives signals in its dendrites and it receives enough excitation to generate an action potential, the inside of the cell will go from negative to positive and back down. And so, as you can see on the right here is a recording of this action potential. So, the inherent negative charge found in the neuron is called the resting potential. And this varies between neurons. And I know in all my classes I hear different numbers, but it's usually on the range of -80 to -40 millivolts. And as I mentioned, the action potential will fire in an all or none fashion, which means that there is a threshold for the initiation of the action potential. And so, as I mentioned, the axon hillock, where the cell body becomes the axon, is involved with being able to sense this threshold. So if you were to stimulate this neuron so that it became slightly positive, but we're not able to stimulate it enough to the threshold, it would not send a signal. But if it receives enough stimulation so that it reaches this threshold, it will send the action potential in all or none. And so that will cause sodium channels, which will allow positive ions to come into the neuron so that it will actually become positively charged relative to the external environment. And in order for the neuron to continue to function, it cannot stay at this positive charge indefinitely, but it must be able to return to its resting potential. And so, to do this, the neuron will open potassium channels, which are another form of positive ions, but that naturally have a high concentration inside the neuron. And so, as mentioned earlier, diffusion will cause particles of high concentration to go to areas of low concentration. So since there's naturally a high amount of potassium inside the neuron, when those channels are opened and they're able to bypass the barrier of the cell membrane, they're able to leave the cell. And since a positive charge is leaving the cell, the neuron will go back down to become less and less positive and more negative. And interestingly, this will actually cause the neuron to become more negative than when it was at its resting potential. And this is known as hyperpolarization. And to be able to return to resting potential so that it can send a signal again, the neuron has a special structure called the sodium-potassium pump that will use energy to excrete sodium and to bring potassium back in. So it's able to bring itself back into equilibrium, and so that it is able to repeat the process over and over again. And I have no idea how often this happens a day, but it's gotta be at least like millions or billions of times a day. Especially if you consider all the neurons in your brain that are constantly firing, constantly receiving signals, and constantly affecting the way that adjacent neurons will function and communicate with each other. So, as mentioned, these neurons are not physically linked. The brain is not one like whole structure. It's actually just very little subunits, very small cells that will all form networks with each other and are suspended in extracellular fluid and supported by glial cells. And so, because they're not connected to each other physically, they must communicate with each other through the chemical signalings mentioned earlier, known as neurotransmitters. And so, because there is a very small gap between the presynaptic terminal of the neuron sending the signal and the postsynaptic dendrite receiving the signal, this diffusion is able to occur very rapidly. So you can see that there's high concentrations of the chemicals found in these little bubbles called vesicles that can be excreted and diffuse quickly to basically saturate all of the receptors here so that every receptor is receiving the signal. And these act for a very short amount of time but have very powerful impact. In fact, the signal is so powerful that as soon as it's sent, the brain needs to rapidly degrade it and stop the signal. So there are many different ways for stopping the signal. As I mentioned, you could break down this chemical so that it no longer fits the receptor and affects the postsynaptic neuron, or you could also recycle it. So the neuron itself could have channels for taking this neurotransmitter back up into it, which it can then repackage and resend. And another mechanism is that the neurotransmitter can actually just like float away and no longer be able to bind to the receptor. And so all of these play a critical role in regulating the signaling in the brain. If some of these mechanisms were damaged, you could have uncontrolled activity in the brain, which could lead to epilepsies or just this hyper signaling, hyper electrical activity that can cause damage to the brain and can prevent you from properly functioning. And another pretty interesting thing is that psychoactive substances like pharmaceuticals can actually mimic the neurotransmitters. So it can do this by either copying its structure, like having a structure so similar that it's able to bind to the receptor, or it can act on the presynaptic neuron and stimulate it so much that it will release too much of this neurotransmitter. Another mechanism could be that it could block the stopping of the signal. So an example of this could be antidepressants, which the most common one now are known as selective serotonin reuptake inhibitors. And what these do is they stop the recycling of the neurotransmitter, in this case serotonin. And so if you stop the stopping of the signal will be there longer and have a greater effect. And this is hoped to help combat depression. And there are many other drugs that can affect the brain in less positive ways that can either increase the signal, as I mentioned, which is known as an agonist, or it can actually act as an antagonist. And this means that it can stop this signal from being properly sent. Oh, I don't know what happened here. Sorry about that. And so one of the most common antagonists that I feel like you will all resonate with is caffeine in your coffee and tea, soda. And what this does is it blocks a neurotransmitter called adenosine. So it will bind to this receptor in the dendrites and prevent it from receiving the adenosine signal. And this adenosine, its normal function is that it makes you sleepy. So caffeine will block the sleepiness signal, and it will cause you to be more awake because your parasympathetic nervous system, which is involved in relaxing you, is less active. And that's often why caffeine can make you hyper, because if you're only receiving signals that make you more awake and not receiving the ones that will calm you down, you can just become overactive. And so now, does anybody have any questions about the neuron or anything separate?
So when signals are too slow, that can cause degenerative diseases. Like what?
So I mentioned amyotrophic lateral sclerosis, but I believe multiple sclerosis is also caused by degeneration of the myelin and signaling that's too slow for your brain and nervous system to properly function.
Okay, so then when the signals like aren't controlled and there's like too much going on, that can then cause like epilepsy.
Yeah, so if there's too much brain activity, that can lead to like seizures or epilepsy. So it's basically just uncontrolled activity of the brain, which can lead to damage.
Okay, so like what would experience like that?
Yeah, so one thing that's very important in the brain, and it kind of seems like all aspects of life to me, is that you really need to find a happy medium. You can't really go from one extreme to the other, because if you have too little signaling, then you could go into a coma because you're not able to stimulate yourself. If you have too high signaling, you could develop psychosis and not know what's real, or you could have seizures. So the brain, the main function of it, and the reason why it has all these different ways to regulate itself is because it needs to maintain a balance so that it can have proper function.
Isn't Parkinson's also caused by the destruction of myelin?
It's dopamine.
Dopamine.
Yeah, Parkinson's can be a result of damage to a part of the basal ganglia called the substantia nigra, and the reason is that it's involved with the execution of movement. So I'm not sure if it's the myelin specifically or just the entire neurons that will degenerate and die so that it can no longer function, and that can lead to tremors because it's not able to properly regulate its movement. Yes?
Do you know anything about like Alzheimer's and dementia and how that relates to neurons?
Yes. So Alzheimer's, from what I understand, it can often begin in the hippocampus, but it can spread to the entire brain. And so there are two main anomalies found in patients with Alzheimer's that are critical areas of research right now. And these are neurofibrillary tangles made up of tau proteins and also beta amyloid plaques. And so the neurofibrillary tangles are found inside the neuron, inside the cell body. I can try to pull that. Yes. So it would be found here in the center where the nucleus is housed. And there seems to be either like a mutation or some sort of dysfunction in these tau proteins that I believe are naturally involved with the formations of microtubules, which are basically railway systems that allow for transport of proteins that are made inside the cell body down to the axon, where it could either be excreted as a neurotransmitter or act as a receptor or a channel. So I believe the tau are made from that, and then the beta amyloid plaques are made from this precursor protein. It's called the beta amyloid precursor protein, which is basically the protein before it's been processed. And normally it will get cut in half to have some function that will help the neuron, but either through like a mutation or some sort of external signaling that messes up that process leads to these proteins that are very sticky to each other, and they're actually able to associate together so that normally they're like dissolved inside the liquid of the cell known as the cytoplasm in the cytosol, but they begin to like clump together and form these plaques, or these like big balls of just protein that disrupt the functioning of the neuron. And just these small changes in the functioning of these normally helpful proteins can lead to improper functioning of the neurons, either through like improper signaling or improper receival of signals or some other factor, because there's so many, that actually causes the neurons to die. Because if the neuron's not receiving any signals, then it will actually kind of commit suicide because it doesn't really have a function. And it needs to receive these growth factors and survival factors that accompany the signaling to properly maintain itself and to prevent apoptosis, which is the cell death, cell suicide. Yes?
Do you know anything about the relationship between the flashlights and epileptic seizures?
Flashlights and epileptic seizures? I'm not entirely sure. I probably just know as much as you, where like strobe lights can induce seizures. I'm not entirely sure the mechanism of that, but if I were to try to make a guess, it would probably just lead to just hyperstimulation of maybe the occipital lobe and the visual centers, and maybe like an odd pattern of the signaling. And the brain is obviously like predisposed to this, where it could maybe act like as a trigger to then, like if you're receiving this odd pattern of signaling from this one neuron that's sending the visual stimulus, and then it will spread out to all downstream neurons and create this, I'm just guessing, so I'm not entirely sure, but create this abnormal pattern that just spreads out and leads to over excitation and unregulated signaling, which can lead to seizures.
Very impressive. So we think that the human being is tremendously complex, and what you talked about is this little 3-pound mass that really runs everything and coordinates everything. And thank you so much for your knowledge and experience. And what we'll do is take a little break, and I'll bring the pizza in the back, and then we can talk a little bit about either some more questions or the music research team, and invite you folks to sign up if you haven't already. And again, this will be contact. It's not like a commitment that you have to do anything, but just that when we have a meeting, we'll let you know, and if you want to come, you're more than welcome. And if somebody didn't get the How Music Heals the Brain, by all means, please take a copy. Thanks again, Kevin, and we'll continue the discussion in just a moment.
And thank you all for coming, I really appreciate it.